Hi, everyone. My name is Dr. Sue Cook, and I'm a lecturer of biology and biotechnology at UNH at Manchester. And I'm also faculty advisor to Team Cook, the student team that is currently working on the NOMADS, Novel Methods of Antibiotic Discovery in Space project. And I'm here to tell you a little bit about what they're doing. NOMADS is a project developed by a team of UNH Manchester students that was submitted to the Student Payload Opportunities with Citizen Science program sponsored by NASA last fall. And their goal is to examine the effects of the space environment on soil bacterial mutation rates and the impact of that on antibiotic production by those bacteria. The NOMADS project is going to investigate the impact of space on mutation rate of soil using a novel device called an iChip. An iChip allows us to keep our bacteria in what we call an in situ like environment, meaning that they're kept in contact with the original soil from which they were obtained, so that that soil can provide the nutrients and environmental conditions needed by the bacteria and hopefully allow us to capture the maximum amount of diversity from the soil sample. The iChip and the soil will be contained in a 3D printed 3-layer box designed by the team that's been produced in-house at UNH Manchester. And then the whole thing is housed inside a box called a NanoLab provided by NanoRacks. So we were very limited on size based on the size of the NanoLab. All of this will then be transported to the International Space Station sometime in spring 2022 for a 45-day expected deployment. So during that time, the bacteria will be exposed to the ambient environment experienced by the astronauts. So it will have microgravity and all the ambient temperature, pressure, and other variables experienced on the ISS. And there will be no interaction of the astronauts um, with the iChip or any of the bacteria during that time. It's just literally being exposed to that environment. And then subsequently, it will be returned to us so we can examine the bacteria and compare them to those from an identical setup that we have that will remain here on Earth. So we can look at the difference in the mutation rate of the bacteria in the two experiments, and also look at the percentage of those bacteria that are producing antibiotic compounds, and specifically the percentage of those compounds that are novel. There's obviously a lot more to this project that I would love to share with you, but we're limited on time. So if anybody has any questions for me, I would be glad to answer those.